Okay, for question 1, draw the structure of an antibody. So as we can see here, I've drawn the structure of an antibody. This is a good way to draw it because it shows the variable region, constant region, complement binding site, heavy chain, light chain, N-terminal and C-terminal, including your disulfide bonds, like exactly where they are. So this is a very good way to draw the antibody. As for explaining the antibody that you've drawn, you want to start off with explaining that firstly, antibodies are proteins. Proteins have an N-terminal and a C-terminal. N-terminals consist of the amino group, while C-terminals would consist of the carboxyl group. We can also see I've labeled the variable regions here and the constant regions here. The variable regions is going to be what binds to our varying antigen, and the constant region is going to be what undergoes isotype switching. It's also important to note that antibodies are held together by disulfide bonds, as I've drawn here, and you also have a heavy chain, which is whatever I've circled in the middle, and a light chain for whatever is adjacent to that. You also need to note that it's a complement binding site for binding of complement proteins, especially C1 protein, and you also have a FC portion which binds to FC receptors depending on the type of antibody you're talking about. So how many types of antibodies are there, or what types of antibodies are there? There's 5 main types of antibodies: IgM, IgG, IgA, IgE and IgD. These antibodies are made as a response to interleukins and interferons. For example, IgG is produced as a response to interferon gamma. Okay, it's produced as a response to interferon gamma, while IgE is produced as a response to interleukin 13 and 4, and IgA is produced as a response to TGF beta. Okay, so each of these antibodies have varying functions. As for IgM, it is the most different from all the antibodies. It's the biggest antibody with an extra 20 amino acids on its carboxyl terminal. It is also the first antibody to be produced by B cells, and it functions for complement activation and agglutination, which should make sense because IgM is secreted as a pentamer connected by a J chain. As for IgG, it has similar functions to IgM, including complement activation and agglutination, but in addition to that, you also have opsonization and neutralization. IgG is also the only antibody to cross the placenta. This is very important because this is a form of naturally acquired passive immunity. As for IgA, this plays a big role in our mucosal immunity. Hence, we'll find it in our mucosal membranes, tears, saliva, things like that. You'll also find it in your breast milk, which is why it's also a form of naturally acquired passive immunity. As for IgE is going to play a role in your allergic responses, and it also functions as protection against parasitic and worm infections. As for the role in allergic response, I will explain subsequently. As for IgD, there is no clear function of IgD, so I won't be going through that. Now for the third question, the role of B cells and mast cells in allergic reactions. Okay, so firstly, what is an allergy? An allergy is a response to an innocuous antigen, and this antigen can come from anywhere, but is most of the time inhaled. So when we inhale the antigen, or if it was to get into our circulation by other means, it would encounter our sentinel cells, mainly our antigen presenting cells, macrophages and dendritic cells. Dendritic cell and macrophages would then process these antigens by phagocytosing them and mounting them onto MHC2 proteins. And these antigen presenting cells would then move into the lymph nodes and present the newly acquired antigen to T cells. And this can go one of two ways. You can get a Th1 response or Th2 response. Th1 response is a protective response producing IgG which eventually binds to mast cells. Our allergic response is going to be a Th2 response. And what determines a Th1 or Th2 response? Well, it's going to be what the antigen presenting cells secrete. So if the antigen presenting cells secrete interleukin 10, you're going to get a Th2 response, which is the case for allergic patients. As for Th1 responses, it's going to be caused by interleukin 12. So we're going to focus on this since we are looking at allergies. So once we get Th2, T helper 2 cells are going to move into the circulation where they encounter B cells. They are then going to signal the B cells to isotype switch from IgM to IgE. This is going to be done, as I said earlier, by interleukin 13 and interleukin 4 secreted by Th2 cells. IgE will then go ahead to bind to mast cells, and this is called our sensitization phase. If we were ever to come into contact with the same antigen again, the antigen would bind to the IgE bound to the mast cells Fc epsilon receptors, and that would cause mast cell degranulation, leading to a release in histamines and prostaglandins. Mainly, you would have your immediate response, your over minute response, and over hour response.